Thank you, Eddie, and thanks for the nice introduction, for having me here. So we're working on a new revolution and trying to learn how to write the genetic code after spending a while reading it. But let me just orient you real quickly in terms of this is the JCVI campus in Rockville, where we have 5 buildings. We've recently expanded to La Jolla. So I've seen the light like Eddie and spending more time on the West Coast now. We've designed and hope to start construction this year on what we think will be the world's first zero-carbon laboratory building, and this will be built on the UCSD campus with a nice view of the ocean. Also in La Jolla, we have the company that Ham Smith and I started, Synthetic Genomics, and the work I'm talking about came from all these different campuses. We've been trying to ask big questions. Everybody asks some of these questions in science of what is life, usually after smoking something. Not as many people asked if they could digitize life. Lots of people have asked how extensive is it? Reductionist biology is, can we pare it down to these basic components? I think a relatively novel question is now, can we regenerate life out of this digital world that we've all been creating? So let me walk you through it. Obviously, digitizing biology is what most of the people in this room have been doing, and what the field of genomics has been doing for a while, reading the genetic code, going from this analog world of DNA into the digital world of a computer. We started this on scale with ESTs in the late 80s, early 90s. I haven't had a chance to look at the database this year, but about a year ago they jumped up to over 50 million EST sequences in the best. So that's a lot of digital biology right there. And 14 years ago, we sequenced the first entire genome of a living species. It's amazing that was in reality only 14 years ago. We now do multiplexing of 8 genomes per 454 run, and so it puts this accomplishment into kind of a tough context. Everybody knows there was a huge expansion over the last decade, starting with the 3 different domains of life, with first bacteria, then archaea, then yeast, and going up in a huge number. I'm humbled to say that this institution here, the JGI, has now done more genomes than any other institution, including the Venter Institution. But together, JGI and the JCVI have accomplished about half of what's out there in the world. So I think it's a nice combined record. 2 years ago, going up to the first digital human genome. And the goal with all these new technologies is going to be expanding this tens of thousands of fold over the next decade. And I think looking at variation in human and applying this to human biology is going to be a big challenge. Right after the first draft of the human genome in 2001, we started looking at the environment more broadly. Those of you who are microbiologists know that the microbes are the majority species on the planet, and we're just kind of either a source of food or a minor irritant to them. How extensive is life? We've been defining that by applying the same shotgun sequencing techniques to the environment, first with the Sargasso Sea, and then expanding that to the human biome, where we found that we have maybe 10 million genes associated with species that are integral to us and internal to us in many cases, in contrast to our fairly dismal number of genes. Applying this broadly to the oceans with the Sorcerer expedition, we a year or so ago completed a circumnavigation taking samples every 200 miles. Last summer, going from Washington, D.C. back through Panama up to Glacier Bay to take samples off of melting glaciers. We've now had 3 expeditions to Antarctica, not on Sorcerer, but by Australian ship and helicopter. And the sorcerer just left San Diego on Saturday for a 2-year sampling expedition, starting with the Baltic Sea, then the Mediterranean and Black Seas. The challenge is we all try to fill out these databases of a gene catalog of planet Earth, is we have to look in more and more unique environments. These are all inland bodies of water that only have minor flushing out to the ocean, and they've been surrounded by early humanity for quite a while, having a huge impact on what's there. So we expect this uniqueness that we're finding of 85% of these sequences being unique every 200 miles to very much continue. Doug Rushus here has developed a technique that a lot of people are using now with the fragment recruitment plots. And just this sort of summarizes quickly what we found that I think surprised essentially everybody. We were told, for example, in the Sargasso Sea there was only going to be a few organisms because there was no nutrients and nobody could find much out there. But what we've been finding in the oceans is this tremendous diversity under a 16S ribotype. So if you were a 16S RNA person, you would say this was a single species, yet we find 40 to 50% sequence divergence, even though the gene content, gene order is largely the same, except these plots can pick up nice variable regions in different genomes. Sometimes a blank spot in the fragment recruitment can be an integrated viral genome, as an example. Now, as we get more and more data, and we're up to around 20 million sequences now, instead of getting more complex, the picture's starting to get simpler. So in fact, if you measured just ribotypes, there's these 5 related taxa that represent 37% of all the sequences we find in the ocean. But underneath these, we find this tremendous sequence divergence. If you look at all mammals varying by maybe 10% sequence divergence, and you look at 40 to 50% in these are almost anti-Darwinian thoughts that instead of survival of the fittest, we have this divergence that's occurred over a very long period of time with all the organisms sticking around for one reason or another. We've now doubled the number of genes at least 3 times. When we did the first sampling in the Sargasso Sea, there were roughly 1 million genes in the public databases. That paper added 1.3 million. The PLOS issue added 6 million, and the team's working on a description of the 20 million gene set. The trouble is the computational tools and challenges get harder and harder at each stage, but we're likely to, over the next 2 years, just with the next sampling, double this number again. And the challenges of looking at this diversity are huge. Are we still finding new things, new gene families? Shabu Yusuf's done this work of basically assembling all the sequences in our data sets with everything in every public database we can find. And basically, if you're looking for new mammalian genes, stop, because basically they've all been found. What you'll find is a lot of diversity. Like if we sequenced everybody in this room, we'd probably find a few thousand variants in each gene. But if you're looking for archaeal viruses and bacterial sequences, we're basically in a linear stage of discovery. So can we pare life down to basic components? Obviously, that was the first thing many people thought when we saw the first genome. And the second genome we did that year was Mycoplasma genitalium, which only has very few genes. It's still the smallest genome of a self-replicating organism, 482 protein coding genes and 43 RNA genes. And we simply started asking questions: if one species needed 1,800 and the other only 500, was there a smaller subset that we could define? And we spent a long time trying to define this in various ways, by knocking out genes, by doing comparative genomics, trying to find out how many are essential. But the tools are so limited that you can't do 100 sequential knockouts in this species. And we finally came down to the choice of the only way to answer these questions was to synthesize the genome where we could directly vary the gene content. As soon as we made that decision, it opened up a number of technical and ethical challenges. Could the chemistry permit even making these large molecules? And even if we made a large molecule of DNA, could we boot it up, or were the barriers to that way too high? We had some very slow starts. The synthesis of DNA, everybody in this room I'm sure knows, is an N-1 situation. So the longer the fragment you make, the more errors you accumulate. So even though something like Phi-X, we thought we could just make 50 base pair oligos, overlap them, PCR them up, and get the genome. We actually got pieces the right size. But even with a selection by infectivity, which gives you about 1,000,000-fold selectivity, there wasn't a single correct genome that would boot up. So the team, primarily Ham Smith and Clyde Hutchinson, developed all these error correction methods that allow us now to go from the digital world and the computer to making pieces on the order of 5 kb quite robustly. So we had things that were all 100% what we designed. But an important thing I want to keep stressing is you can only build something as accurate as what's in the public databases or in any database. It's interesting, Clyde Hutchinson was in Fred Sanger's lab when they sequenced Phi-X, and he stuck a sample of Phi-X in his pocket that he's been carrying around all these years. So we were able to resequence the original sequence, and there were only 3 differences out of the 5,000 base pairs. None of them would have been lethal. So what the last speaker just referred to, the Bermuda standards. The Bermuda standards are incredibly unsatisfying and will not allow the field of synthetic genomics to take place. It's a very low standard of quality that is fundamentally non-tolerable going forward. The fun stage was when we took this 5 kb piece of DNA and stuck it in E. coli. E. coli recognized it as normal DNA, started making proteins that self-assembled to form the virus. The virus was not very grateful. It turned around and killed the cells. That's how we detect them. This is a cartoon of what Phi-X looks like. We call this the software building its own hardware. Lucy was using computer language terms. We use those extensively as well. And I think this is a very important concept. While computer people would love this to be the case with computers, it is the case with biology. Our goal was not to make a 5 kb virus, it was to make bacterial genomes. We thought we could do this by making viral-sized cassettes of genes and then putting them together, but this became a huge challenge. Again, trying to make the Mycoplasma genitalium genome, we had to start with what was in the digital world. So in 1995, when we sequenced the genome, the standard was 1 error in 10,000 base pairs. We figured that was not good enough, so we resequenced the genome and we found on the order of 30 errors in the sequence, many which we think would have been lethal had we made that sequence. So we started with new digital information. And started designing the smaller pieces that would overlap to make the bigger ones, that would overlap to make the larger ones. When you're doing work like this, you have to worry about artifacts, the biggest artifact we were afraid of is one single molecule of native genome as a contaminant could get booted up and fool us to thinking that we had been successful. So we decided to alter the genome extensively with putting DNA watermarks in. And some people have complained that we didn't put something really profound in the genome, like one small step for microbes. Ham, Clyde, and myself and our colleagues just signed the genome. But it's, if you let your imagination go, so we have a 4-letter code, triplets code for 20 amino acids, all with a single letter designation. So you can write all kinds of interesting things in the genome, and we're trying to be much more clever with the new genome. We're synthesizing now to have codes within codes, because everybody solved the last one too easily. But it's also important in terms of identifying this as synthetic DNA, identifying the lab that produced it. If resulting organisms get out, they'll be traceable back to their source. I think these are not just fun criteria, they're really important ones. Now, we started assembling these in vitro. We would put 4 of the sub pieces together to make 24 kb pieces. We cloned each of these in E. coli, produced a lot of them, sequenced them, because we're trying to establish new techniques with really high fidelity. And it worked pretty well up until we got over 100 kb. And then only about half the fragments cloned readily. And we started looking around for different systems. We had a couple of postdocs working for several years on characterizing the recombination system from Deinococcus radiodurans. This was one of the early genomes we did. In fact, I think the first one funded, well, Mycoplasma genitalium was the first one funded by DOE. I think this was the first official DOE genome funded by DOE. They were worried that Mycoplasma might be a pathogen and they'd get in trouble with NIH for funding it. But it's characterized by its ability to take huge doses of ionizing radiation. You can see in the top panel, after 1.75 million rads, basically 200 double-stranded breaks, the genome gets blown apart. But somehow, 24 hours later, it's able to completely and reproducibly reassemble its chromosomes. We kind of like that. We view this as the biological equivalent of our genome assemblers in the computer. And this is exactly what we want to do with small oligos. But we could never get this to work even with all the repair enzyme systems in vitro outside of Deinococcus. But in the process of investigating species, it turns out everybody's lab favorite, Saccharomyces, does this very nicely on its own. We can actually clone very large pieces up to, maybe there's no limit, but at least on the megabase size. And we found that we can use yeast homologous recombination system just to assemble the pieces. So we had the quarter molecules, which were about 175 kb. We put these in a vector in yeast, and yeast automatically, within a very short period of time, assembled the entire genome for us. So this was quite exciting. We pulled it out and sequenced it. It was 100% accurate of what we had designed. And this is the largest molecule of a defined structure been made to date. Two scientists, Dan Gibson and Gwen Benders, were key to making this happen. So the question is, well, it could do these large quarter molecules. Could it do even smaller molecules? This is just a size comparison of phi x up to Mycoplasma genitalium and what has been synthesized. So this is substantially larger than any piece of DNA that has been made to date. You can actually see it. It's a circular molecule. You don't need an EM to have it show up. But we tried this with a smaller set. And we don't know yet how small we can go, but we recently published this in PNAS, putting 25 sub-pieces into yeast. Yeast took them all up and assembled them into the genome. So this takes a process that was taking us years to do, to the point that we can do a very large number of genome assemblies per day just by cloning the pieces in yeast. So it's extremely powerful. These processes, you can find them in the paper, were very well characterized. So the challenges, we met one of them. You can make large pieces of DNA very accurately. Can you boot them up? I was pleased to see Lucy using the same terms for what she was discussing. And I think this study that we published a couple years ago in Science is one of the most profound ones to come out of my labs, my institute, when you think about the implications from it. Simply by changing the software in the cell, we converted one species into another. The only thing that changed was the DNA that was in there. So let me walk you through that because the implications for this and the next stages are very important. We had two related mycoplasmas, roughly the same distance as human and mice are apart. We isolated the chromosome from M. mycoides. We treated it partially with a number of proteinases because the challenge was to find out would naked DNA actually work, or were there going to be some proteins that we had to have to get a chromosome booted up. We had a couple of gene sets to it, one for selection and one so if the chromosome was booted up, it would turn the cells a bright blue. And then we inserted the chromosome in the recipient cells. I'm glossing over a lot of details. As many of you know, you can't pipette large pieces of DNA like this. The team worked out how to move these around in gel blocks, how to, and keeping the chromosomes totally intact with no nicking, etc. So we're not 100% certain what happened, and we're starting to dissect all these processes. We had the sequence of both of these genomes. What we think happened when we inserted the mycoides chromosome into the capricolum cell, there was a brief period where we had both chromosomes in there, and one of two things happened. One is the cell went through a division and the chromosomes just segregated to daughter cells. A more interesting idea is that this new piece of DNA started getting transcribed, including the 3 restriction enzymes that were in there, that we knew these restriction enzymes recognized the capricolum genome. We think because of one possibility of what was transcribed first, that they recognized the capricolum genome as foreign DNA and chewed it up. And in a short while, we had cells with only the mycoides chromosome in it. They became bright blue cells. When we sequenced the proteins, there was no hint anymore of capricolum proteins, the lipids, everything that was defining that one species was gone. And everything that now defines the mycoides species was present, and the only change was the DNA software. When we tried this with the synthetic chromosome out of yeast, it didn't work. So it was the same sequence. So we worked out proteins were not required, but something else obviously was. And we have a couple papers that have been submitted that I can give you a quick synopsis of how we've been breaking down the principles of what can happen. So we can take chromosomes, bacterial chromosomes, and grow these now as artificial chromosomes in yeast. We have, in fact, one yeast strain where we have 2 bacterial chromosomes growing independently in it. We can take these out in our agarose plugs, and if we transplant that genome, the mycoides genome, into a capricolum strain that we made where we remove the restriction enzymes, that transplant actually works. If we methylate the chromosome in vitro in these plugs, and the technology associated with that was some tremendous challenges, and then we deprotonate it, that then transplants into the native capricolum and protects us against the restriction enzymes. So these are key steps now that allow us to go from a bacteria, take the chromosome out, grow it as an artificial chromosome in yeast. Those of you who work with different species without genetic systems, we now have a genetic system. It's all the yeast genetics, and we can make any change that you can do with a yeast chromosome. We can now use those tools for the bacterial chromosome, modify it and transplant it in bacteria and make a new species. So you don't have to make a synthetic chromosome. We can now, using homologous recombination, all the tools in yeast, do this complete cycle. And I think that is likely to make some important changes in some of the next steps. This audience is probably not one that asks this question, maybe it is, but lay audiences ask this question much more often. Why do this? You're scaring us. Some people have already addressed this at this conference. Obviously, we're going from, in theory, 6.5 billion people to over 9 billion people over the next 40 years, when we can't sustain very well the 6.5 billion we have. When countries do develop, there's an exponential increase in their energy demands at a time when, while oil companies are all saying they have sufficient reserves, they do at the moment when the economy is down, but I think it's pretty clear cut they're not long-term oil reserves. We need, obviously, to cope with that, but I think the single biggest issue is last year the 4.2 billion tons of CO2 we added net to the atmosphere. So we're trying to see if some of these tools we have could also be applied to these problems. So where do we go from here? We view these 20 million genes we've discovered to date as design components. If you look at the electronics industry in the 40s and 50s, there was only a couple handfuls of design components: resistors, capacitors, transistors, eventually integrated circuits. Right now we have 20 million design components. Some of these, if you look at things like ABC transporters, we have 50,000 different ones. Some gene families, like the photoreceptors that we're finding ubiquitously throughout the ocean, the same as our visual pigments. We now have tens of thousands of those, many of which see dramatically different wavelengths of light. So think of these as design components. The number and the variety is only going to go up. We now have software at Synthetic Genomics for designing species. And wouldn't it be great if we could do all of this on first principles? But basically, the only one that's really working on first principles is Lucy's lab, and we need some of those circuits that she's working out. We're trying to just build cassettes that we know work, not necessarily knowing why they work. So we can choose metabolism. Is it CO2 to methane? Is it glucose to lactic acid? We can just build these as cassettes from PCR cassettes and design things. Importantly, with people concerned about the environment, as we are with suicide genes, autotrophic markers, ways to really regulate these organisms and keep them under control. Possibly viral resistance. If you're thinking of producing large amounts of things, normal biology doesn't work. We're defining this new area I'm calling combinatorial genomics, because if you think about trying to even enhance a 10-gene pathway, and you have 10 different versions of each of those genes, that's 10 to the 10th variants you got to come up with. Well, as I said, we have tens of thousands of variants of many genes, and that's only going to go up. So now with this homologous recombination in yeast, we can take cassettes, either synthesized cassettes, PCR cassettes, and we think we can get up to making 1 million genomes a day. And then we can do biology like it's happened since the beginning of selectivity. So it's a different level of screening and a different pace with evolution. While teams are working out how to do this on first principles, this may be the mechanism where we discover most of the first principles in a retrospective fashion. We've been working on what we've called fourth generation fuels, trying to use carbon dioxide as the carbon source for the fuels and sunlight as the energy source. We've been studying secretion pathways, and the team has come up with a new algae that instead of just accumulating lipid, they secrete whatever lipid size we want to engineer. So they can secrete pure C8, pure C10 mixtures. You can go along the entire pathway. So this changes algae from what everybody was looking at as a farming problem to a manufacturing problem. So we're trying to get algae to grow in a continuous production mode, pumping out these lipids. They come out in a pure form, and they can just be accumulated on polystyrene beads, or literally creaming the skim, or skimming the cream off the top. Also, the third genome that we did, the first archaea, got me thinking about these pathways from early on. And people have studied methanogenesis long before have thought about these issues of, here's an autotroph that takes CO2 to methane. And we've been working on a program with BP looking to use biology to convert coal into natural gas. And part of this was just using the same environmental genomic tools to discover new organisms deep in the earth. And using, we have, as far as we know, the world's only anaerobic cell sorter. We can sort cells from an anaerobic environment into microcapsules, and we have about an 80% now efficiency for culturing these. So we have now a large number of organisms that live off of coal as their sole carbon source. And they break down the coal into a series of organic acids. We have in these environments 2 unique archaeal methanogens. Ones that eat the organic acids and produce methane and CO2, and others that eat CO2 and hydrogen and produce methane. And it turns out the digestion of the coal is done probably by secreting a very large number of enzymes into the coal, which metabolize it into these organic acids. Turns out coal is ancient plant material, as you know. All these enzymes look like they might do it even more efficiently with plant material. So all these things are coming full circle. Putting a few of these different species together in a defined culture, we can get large bubbles of methane gas just coming off a small amount of coal. And we look at the rates that this happens and applying it to the subsurface, we think we can convert huge areas of deep earth coal into natural gas, and BP is one of the leaders in just pulling that natural gas out of coal beds. So we see a lot of potential for synthetic genomics, starting with that digital code in the computer, 4 bottles of reagents, and designing and making what you want. I think within a decade, if not sooner, people will be designing novel plant species with the characteristics that we need. With these new combinatorial methods, the kinds of shifts that we've seen with rapid DNA sequencing and what it's done to the information explosion, I think we will have a chance to now maybe do this on the biology front, and we're applying it to some unique areas of low-hanging fruit of trying to capture back CO2 and make fuel. But I think the most profound effect will be on the broad understanding of biology. That's my tale, and I'm sticking to it. Thank you.
Questions for Craig? It's a good tale. So have you put the synthetic coming off oligonucleotide synthesizer machines assembled genome into, have you got it to boot?
So we are making a new chromosome from a species that we know will work for sure. But we made a bad choice by trying to go with the smallest genome, and by being the smallest genome, it grows so damn slowly. It takes 6 weeks to do each experiment. So we're actually making a much larger chromosome. We think it's not going to be very exciting. I know the world will view it as an important step, but the important steps are these, because we now think it's totally obvious that it's going to happen. But no, we have not done it yet. We expect it by summer.
So what I'm thinking is, if for a computer to work, and you have to have every part work perfectly. And now if you engineer a software that put into the, like a bacteria, and it put into the environment, which is not, everything is not perfect. And so then maybe that will imply you don't know the outcome of the machinery you're creating. So how do you envision that will impact the environment?
Well, number one, there's no intention of putting these into the environment. I think that would be ethically wrong and biologically really stupid for some of the reasons that you suggest. So I don't think anything should be built in the laboratory that could survive outside the laboratory or outside a production facility. I think that's critical for this next step. We're trying to work out laws and rules and regulations. I think that's the single most important one. And it's a little bit worrisome because there's about 150 algae companies just from the last year, I think, in the Bay Area alone. And many of them want to use recombinant algae in open pond systems. My personal thought is that should not be allowed unless they're under such tight autotrophic regulation, suicide gene control, that they would not survive without unique chemicals. Some of the algae species, so in my home in La Jolla, I have a swimming pool on the hill. When there's a storm at sea, I get all kinds of unique algae species growing in the pool. When we sample the air in this room is full of viruses and microbes. So to think that we can do anything in an open pond environment without extremely careful biological regulation is foolish thinking. So, and hopefully we can define the environments in these closed systems well enough to define the biology associated with them.
If you're going to design a synthetic genome, what are the chances that it works? And are you allowing for some way that the system could make it evolve to make it work?
Well, as I said, we're only limited by our imaginations. You're proving that. So, the initial steps that any of us are taking are by copying nature, right? We started off by building the Mycoplasma genomes and then trying to see what we can eliminate to get down to a minimal operating system. We just heard from Jeff's talk an excellent example of mechanisms for self-evolution. I think we want to be careful with doing that with synthetic species because that could overcome the mechanisms we want to have them be under our control. So while it's obviously doable with a variety of mechanisms, unless we have strict ways to always have, not have them evolve away from our autotrophic markers or suicide genes, we don't want them self-evolving. In fact, for an industrial system, you absolutely don't want them self-evolving. You want, if anything, to limit evolution.
Okay. Ham Smith and Clyde Hutchison, they found an operon regulator in the mycoplasma and published it. And of course, operons were discovered in 1961. Do you think that it might be a good time now to revisit the operon regulation?
It's always a good time.
Okay, well, we have one final question back there.
Do you think you can make a bacterial chromosome that will boot up in yeast?
Well, the question was, do we think we could make a bacterial chromosome that will boot up in yeast? We're going extremely in the other direction, trying to make sure it won't boot up in yeast, because yeast artificial chromosomes, when they were applied to the Human Genome Project, I think set back the Human Genome Project by 5 years. Because in fact the genes did boot up in yeast and therefore got deleted. This is probably working very effectively because of the codon differences, with other more closely related species, we're deliberately altering the codons to try and keep them from booting up in yeast. I think that's the only way we'll get reliable reproduction, but there's a lot of people here that know more about yeast than I do.
Okay, one final question.
I was just kind of curious, you had that strain at the beginning of your talk, the Orrington Designer 1.0. Well, roughly, what's the, when we get there, roughly how many tens of years can we expect to have something like that?
We talk about the bug or the software?
Both.
Well, we have the software now, and we're using it at Synthetic Genomics to design some of these fourth-generation fuel species. We're not ready to release the bugs or the software because the software has bugs.
Great. Okay, well, this marks the end of the user reading. I want to thank you all for coming. Hope you'll come next year. I want to thank all the